So it's quite exciting to be part of this conference today, and it's actually my first Phage conference post-lockdown, so I'm really looking forward to that. So my presentation is going to be based on the work we have done on Salmonella biofilms, and it's actually part of a bigger project sponsored by BBSRC. And the overarching aim is to develop a phage additive that we can add to feeds of pigs and chickens to control Salmonella. So to do this is for us to be able to assemble well-characterized phages and look at their infectivity and their lysis parameters and host range features as well. And then from there, we can be able to make cocktails. And this cocktail, we can be able to test them in different infection models. We have tested pure cultures and biofilms in vitro. We've also looked at ex vivo models focusing on chicken, pigs or avian cell lines, as well as humans cell lines as well. We've actually done work on Galeria and also including mice model with one of our collaborators as well. And then after that, we'll be able to optimize spray drying parameters. Basically, the essence of this is to formulate the phages into powder form, and then eventually we can add into the feed. So the whole idea is to be able to form these additives that we can add to the pigs and chicken feeds, as I've mentioned. But in my presentation today, I'm going to focus on the biofilm model. So biofilm, as we all know, they are aggregates of bacterial cells on surfaces, which can be biotic or abiotic surfaces. But then the cells form these extracellular polymeric substances, which are rich in polysaccharides, protein, lipids, dead cells, and in the case of other bacteria, spores and other materials. So the essence of this is to see how these phages can be able to remove Salmonella in the biofilm. So I'm currently working for SRUC, Scotland Rural College, but all of the data that I'm going to present today, as I've said, is part of the BBSRC project, and it was done during my postdoctoral role at the University of Leicester. So the outline of my talk, I've just categorized into 5. I'm going to introduce briefly Salmonella and the biofilms, and then the aims of our work and objectives. And then the methods we use to optimize the phage cocktail, the Salmonella strains we have used and how we set up the biofilms, and then the phage treatments and the way we analyze the data. I will show you some of the results we have obtained and also some focal points in the project. So just a quick introduction about Salmonella infection. I'm going to focus on the non-typhoidal Salmonella. So these are zoonotic infections caused by Salmonella species other than the typhi and paratyphi. So they are reportable infections, and the majority of them are foodborne, and they are spread through the oral fecal route. 85%, as I've said, they are foodborne, but then we have other sources such as through pets or even human-to-human transaction, and also through drinking contaminated water. They are associated with very poor press, as you can see from here, dirty meat and Salmonella in eggs in various press outlets. So they are mostly associated with poultry and pork sources. There are several serotypes. So these are ways of typing Salmonella based on the O and the H antigen. And there are also other ways using the phage typing schemes. So several of these serotypes are prevalent and they are common in human infection, but the S. enteritis and typhimurium are commonly found associated with humans. So the infection is quite debilitating. About 94 million infections have been shown globally, with about 21% fatality. And in the EU, it can cost up to 2.8 million euros to treat the infection. Several antimicrobial resistance have been reported within the serotypes, and so the need for phage interventions. So in the literature, there are several individuals and cocktails that have been published using different MOIs to remove Salmonella. And so this work is also shown in vivo with several infection models. And I've put this interesting work just published by one of my former colleague, Anisha, and it's a really interesting read in microbiology spectrum. So I really wish you'd be able to look at this. It's quite, will form an interesting read. So our aim is to be able to apply phages to control Salmonella, and this time in biofilm, as I've mentioned. So our aims are to be able to optimize a new phage cocktail. And to do this, we just don't want to put phages together, but we want to put them in a way that they can complement each other in the mix. So the essence is to be able to improve clinical coverage of the isolates that we are testing, and then to be able to improve the lysis efficacy of the phages. And then we'll hope to look at cocktails that can mitigate resistance development as well during killing. The other aim is to determine the impact of cocktail on biofilms. And so we establish biofilm in different strains of Salmonella, focusing on the infection models that we are targeting, and also the condition we hope to apply these phages eventually. We determine the effectiveness of this cocktail using different methods, which I'm going to elaborate a little bit further in my talk. So the first aim is to optimize a new phage cocktail. So when I say a new cocktail, because initially we optimize a 3-phage cocktail. And so in my graph here, the figure A, we can see the individual phage infection. This is MOI of 10. But then when you put the phages together, 3 of them, you see this complete clearance. Even some cannot be detected after treatments. So these 3-phage cocktail, though they are quite effective, but we optimize them based on 3 representative chicken isolates and 10 swine isolates. And so this is grossly insufficient. So this work was published last year. So we hope to improve on this cocktail. And to do this, we now acquire extra or additional strains from the UK and Thailand. And so we focus on these 2 countries because members of the consortium actually formed from the UK and Thailand. And also these 2 countries are one of the major consumers of pigs and chicken products. So the strains, as we can see here, 36% are from the Thailand and also 64% from the UK. The distribution is quite averagely equal from the pigs and the poultry. So here are the different serotypes that we are able to cover. About 26% of them are these are the different numbers of strains representing the different serotypes. So we did host range assay first using spot test, and we applied 10 to the 8 of the phages individually. So we had 23 phages. Sorry, I've not mentioned that. 23 phages, 22 of them are myoviruses, and 1 is virus. So we applied 10 to the 8 of the phages on the surface of the confluence loan of the individual strains. And so we had a very good coverage from the total 23 phages. So about 83%, that is about 140 strains are infected by the 23 strains, while 17%, forming 29 strains, are not infected by these strains. And these are the serovars that are not covered. In some instances, we have complete lack of infection for the different serotypes, and sometimes we have some strains within the serotypes that are covered by the strains. So we put the infection profiles. So the red or 0 is no lysis, and then the yellow or the mustard is 1 with cloudy lysis, and then 2, which is the green. We had lysis on the spots, but then we had some resistant colonies developing from the spot area. And then 3, we had complete clearance. And so we combined all lysis together and look at those strains or those phages that are able to kill maximum number of strains with lysis without resistance and lysis with resistance. So we took the 3 phages I mentioned from the beginning, which we published, and they also formed one of our with the desired parameters. And then in addition to that, we look at those with higher host range coverage as well. And in this case, we are able to look at phage 1, phage 2, phage 4, and phage B7 and 3F. So we added these extra phages to the first 3 phages because we wanted to build the cocktail and look at the interaction. So we added the new phages individually into the 3 phage cocktail and look at the activity and MOI of 10. So we added first individual of the phages, and then different permutations of 2 phage cocktails, 3 phage cocktails, and 4, and then 5 to the 3 phages. So in the end, we were able to, we did several of these killings. So I'm not going to show you all the different killings, obviously, but this is just the final cocktail that we are able to develop. So we're able to develop 5 phage cocktail, that is the 3 initial one with additional of 2. But then in the coverage, there was one particular isolate that only 1 phage could be able to cover. And so we added that in the mix and making 6 phage cocktails. So we compared the 5 phage cocktail and the 6 phage cocktail. And as you can see, there is no much difference in the activity. And so this was done at 37 degrees centigrade and at also 40 degrees centigrade. And with this 6 phage combination, we're able to kill all the 140 that are susceptible to infection by the phages. So the next aim is to determine the impact of the cocktail that we have developed on biofilms. So first, the biofilms condition that we tested. So we had 37 degrees centigrade aerobically, and this is the condition we normally grow the bacteria in. And then the second condition is 40 degrees centigrade anaerobically, and this will mimic the chicks and the pig gut. And then the final condition is 37 degrees centigrade anaerobically, and so this will focus on mice. So I've just added the mice here because one of our collaborators have mentioned will be testing the phages as well. So we added that condition. And also humans, because eventually these strains, though they are prevalent in pigs and chickens, will eventually be found in humans causing the infection. So we wanted to test also this activity. So for the assays, though we optimized the cocktail in the propagating host, which is SC147, and we tested this biofilm in another chicken isolate. So the propagating host is a chicken isolate. So we had another chicken isolate as well. And then the pig isolate, which is the 44SO1, and then a lab strain. So we did a different biofilm assays. We had the 12 well culture plate assays, and this is for us to be able to assay the planktonic cells and the biofilm as well. So in this, we're able to assay the CFU count. We measured the OD, and this is just the OD of the culture's planktonic cells. And we also did this crystal violet staining and did the OD measurements. And then we did colony biofilm. So in here, as you can see, this is just showing a plate with the agar on a plate. And then the black area is filter membrane, and then the little spot here is 5 microliter of the bacteria growing culture. And so with this, we are able to grow the biofilm on the filter membrane without having direct contact with the medium. So we can be able to extract the membrane and assay this. So on all of this, we try to look at the different aspects, although we're not measuring individually, but the cells and also how the matrix are also stained during this process. So I'll just explain a little bit what we did. So initially, we had a growing culture at exponential phase, and then diluted 1 in 10, and that would give us roughly about 10 to the 7 or 10 to the 6 of cells, and that was where we started the measurement. So we did an initial assay, and we saw that the biofilm, maximum biofilm, is formed within 3 days. So we wanted to target established biofilm. So we grew the biofilm for 3 days. And then after 3 days, and that will start our day 0 for the phage treatment, we treated with phage. And during, before then, we refresh the cultures, the biofilm cultures every 24 hours. And after refreshment of the cultures, we will now treat with phage. And after treating with phage, we incubate for 24 hours at the different conditions and then assays the way I've already mentioned in my previous slide. At the end of 5 days, we end the experiment. So the first result I'm going to show here is the planktonic culture. So for the planktonic culture, we only measured the OD because there was so much to do, and we just focused on just to see what happens in the cultures. And we already know what happened in the cultures anyway, because we've already done the work and showing that the phage cocktail can remove salmonella very, very quickly. So we saw, as you can see, the phage treated, so this is all the 5 days. So the phage treated cultures were greatly reduced compared to the controls. And we saw this in the different conditions. But then when you look at it daily, you can see that this is quite consistent as we saw when the results were combined together. So interestingly, for the biofilm, now here we measure the CFU count. So here we remove the planktonic cells, wash the cells, and then resuscitate or recover the biofilm and then measure the CFU count. So we saw here as well that the phages cumulatively for the 5 days showed reduced biofilm measurement in terms of CFU count compared to the control. And we saw this in the different conditions as well, although reduced efficacy was observed in the 40 degrees centigrade anaerobically. When we broke this down, we saw an interesting observation. So for the 0 time, when we started, after 24 hours, the cells were completely removed by the phages. We saw this in the different conditions. So the best treatment condition was a 37 degrees centigrade anaerobically. And as you can see, the phages continue to reduce the bacterium in the biofilm, even up to 5 days. We saw a reduction up to 4 days with 37 degrees centigrade aerobically, and then reduced, much reduced clearance at the 40 degrees centigrade anaerobically. Because even at day 5 for the 2 treatments, we didn't see much a significant clearance. So for the crystal violet stain, so this is another plate where after measuring the CFE count, we had another copy of the plate, but this time we removed the planktonic cells and then stained the biofilms with crystal violet. So for the 37 degrees centigrade aerobically, and anaerobically, we saw a similar result, which I'm not going to show the details of all of them, but for the sake of time, you can see that the phage treatment quite reduced compared to the control. But then with the 40 degrees centigrade anaerobically, we saw increased in the biofilm mass, actually, in the phage treated compared to the control. And we saw this for all the days. So this was quite...
Janet, the time is about to...
Okay, so I will just wrap up now. Actually, it's my second to the last slide. So for us to verify this, we just trying to show what happened, because crystal violet stains both live and dead. And we saw this also in our work with C. diff, where we observed slightly higher cultures, but then most of the cultures were dead, as shown by the propydium iodide stain. So we saw also with the colony biofilm, much reduced cells compared to the control, showing that the phages are actually killing the bacteria within the biofilm. So just to summarize, we show that we are able to develop a phage cocktail comprising of 6 phages, and these have very wide host range properties and effective lysis capabilities, and they're able to reduce resistance development. And then the phages significantly reduced the OD of the planktonic cells, and then removed salmonella, everything non-detectable within 24 hours, and then reducing bacterial load in the different conditions. So future work will focus on we're trying to see what happened on the resistant cells, and also testing the phages in different cell lines and formulation as well. So just to wrap up, just to thank my former PI and all the PIs in the University of Leicester for their great support during my time there. And then most especially, I would like to thank my 2 students, James McDonald and Judy. So they worked with me. It took us 60 continuous days to do this experiment, and they were there throughout. Abdullah helped during the weekend, and that was quite helpful. So I just want to thank SFAM also for sponsoring my attendance here. Happy to take your questions now. Thank you.
Thank you. So any questions from the audience? Yes, there's one here.
Hi, thank you for the talk. I'm Dibyendu from Exeter. Just wanted to ask that while you're trying to do phage optimizations on like liquid cultures to essentially get complete clearance as fast as possible, but does that make sense in terms of delivering it in the organism? Does that optimization still carry over there?
So that's quite an interesting question.
Or something like that would be just better, or yeah.
Yes, so for therapeutic phage development, it's always good to begin with in vitro work, and also then migrate to a more complex environment, so you can get to know your phages a little bit more. So for the phages, we hope to formulate them into powder form, but eventually, even within the powder form, they will have to be released in the powder and then contact the bacteria. So that's why we're trying to test them in different conditions to see how effective they are, mimicking the temperature this time and anaerobic conditions for the prospective organism that we are trying to test. In this case, pigs, chickens, and humans as well. So yeah, it's hard to extrapolate results, what you see in vitro to in vivo models, of course, but then information derived are quite useful in the way you will eventually use these phages in your prospective models. I hope I've answered your question.
There's an online question from
Yeah.
Amos.
Sorry?
There's a question online from Amos.
Yes. Amos?
Okay, thank you, Janet, for a very nice talk. Okay, just to follow up on my colleague, there are these products which normally mimic the gastrointestinal tract environment, like simulated gastric fluid, simulated intestinal fluid. I don't know if you are planning to use any of them, because they're kind of like closer other than using rich media, because in the stomach is something else. Yeah.
Yes, we've tested that in our C. diff work, where we look at fermentation model and, simulating the gut and even spiking with fecal matter from humans and this. So yeah, it's an interesting field, although not part of this project, but I think it will be something really nice to explore. Yeah. And also to say we are testing the phages also in cell lines. This is another concept entirely. So yeah, thank you for that information.
All right, thank you.
Okay, any further questions? Okay, thank you, Janet.
Thank you, everyone.